Welcome to True Projects. In this video, we are going to explain about the project that is Enhanced Crop Disease Detection with EfficientNet Convolutional Group-Wise Transformer. Before getting into the execution, first let us understand what is the project about. Crop diseases. They are major challenge in the agriculture, affecting not just the quantity of crops produced, but also their quality. When diseases they go undetected or they are diagnosed too late, they can severely reduce crop yields, which in turn impacts the food availability and also the farmer's income. Early detection is key as it allows for timely intervention, reducing crop loss and preventing further spread. Without effective detection, resources like water, fertilizers, and pesticides might be wasted, harming the environment and also increasing the cost. Traditional methods of detecting these crop diseases they rely on manual intervention and also the visual inspection by the experts manually. So this can be slow, subjective, and prone to mistakes also. However, we have advancements in the deep learning where we have seen a huge shift in how these diseases are identified. So the models like convolutional neural network and vision transformers, so they were used. Coming to convolutional neural network, they have proven to be good at analyzing images and extracting important features like spots or discoloration on leaves, which are signs of the disease. On the other hand, vision transformer, that is VITs, take it a step further by looking at a bigger picture. They don't just focus on individual features, but also on how those features relate to the entire image. This gives them a more holistic view of the disease spread and nature. So while CNNs are great at zooming in on specific details, VITs excel at understanding the broader context, leading to more accurate and reliable disease detection. While deep learning models like CNNs and VITs have shown great potential at detecting crop diseases, they come with some significant challenges. They have some significant challenges that make them difficult to use the real world agricultural settings. For starters, these models require a lot of computing power, which means they need high end hardware and can be slow to run, especially in places where resources are limited. On top of that, these models often have millions of parameters, making them complex and hard to deploy efficiently, particularly in environments where technology might not be advanced. And the other issue is that these models typically rely on large, well annotated data sets to learn from. However, in crop disease detection, the data sets are often small and not always diverse enough, which makes it hard for the models to learn all the possible variations of the disease. This leads to overfitting, where the model becomes too focused on specific of the training data and struggles to perform well on new or unseen data. This is problematic because crop diseases can vary greatly depending on the factors like crop type, weather, and disease stage. To make these models more effective and practical for real world settings, it is crucial to find ways to reduce their computational demands, handle small data sets better, and improve their ability to generalize across different disease scenarios. So, to overcome with all these issues, this project uses a combination of two powerful algorithms, that is EfficientNet and the other one is group-wise transformer, which forms together as EGWT, that is nothing but EfficientNet convolutional group-wise transformer architecture. First, EfficientNet is considered. It is employed. That helps us to break down the crop leaf images and extract local details, kind of like zooming in on small sections of the image to understand the texture and features. Once these local details are captured, they are passed to the group-wise transformers. They are passed to the group-wise transformers. In simple terms, the transformer it divides the image features into smaller sections or groups. Within each group, it applies an attention mechanism to figure out how the features relate to each other, like focusing on how certain parts of the leaf might be connected or influenced by others. After this, the outputs from each group are combined to form complete picture, which helps the model not only understand the small details, that is local features, but also see how they fit into the larger context, that is global patterns of the disease. This approach helps the system accurately capture both the fine details and also broader disease patterns, making it much more effective for detecting crop diseases. And to train and test the system, here we are using three datasets, that are plant village dataset, cassava dataset, and tomato leaves. These datasets are great because they provide a variety of images of different crops with different diseases, which helps the model to learn to recognize a wide range of patterns. Now, what is cool about the system is is how it combines convolution networks and group-wise transformers. The convolution part helps the model zoom in on important details in the image, like spot or discoloration on the leaves, like and the group-wise transformer then helps the system to look at the bigger picture and understand how those small details fit into the larger context on the whole image. So by combining these two techniques, the system becomes more efficient in terms of how it uses computational resources and how many parameters it needs to process. This is important because it makes the model much more accessible for use in environments where computational resources are limited, like in smaller farms or places with the less powerful hardware. Even with these constraints, it still manages to deliver reliable results, making it efficient tool for detecting crop diseases accurately without requiring a ton of computational power. So to further improve the performance of the project, that is to further improve the accuracy in classifying crop disease, we are incorporating three different models, that is Exception, NASNet Mobile, and XNASNet as the extension of the project. These models are more advanced and they are able to capture the finer details in the images of crop leaves, helping the system understand disease patterns in a more nuanced way. It is like upgrading from a simple camera to a high definition one. So we can see much more clearly what is happening within the crops. And we also added the additional part, that is detecting where that particular disease is on that leaf. So we also have added a detection mechanism using YOLO family of models, which is really good at spotting abnormalities in the leaves. This means not only can the system classify disease, but it can also highlight exactly where the problem is on the leaf. Imagine a doctor identifying not just the illness, but also pinpointing which part of the body is affected. And also we have developed front end using Flash framework. So this makes the system interactive, and we have also added user authentication mechanism to make sure that only authorized users they can access the system. And to implement this project, we have followed the steps mentioned in the flow of work. So let's understand how did we implement. We have followed certain steps for the classification and another set of steps for detection part, and the other set of steps for front end part. Let us understand them clearly. So what we have done it. First, what we do is we import all the necessary libraries and frameworks for data loading, pre-processing, training, and evaluation. For example, TensorFlow, Keras, OpenCV, etc. Then comes exploring the dataset. So here, the three datasets that are plant, village, cassava, and tomato are explored. These datasets contain images of crop leaves labeled with different diseases, which are used to train and evaluate the model. Then comes image processing part. So several transformations are applied to the images to augment the data set, making the model more robust. So what we have done is, first we have adjusted the pixel values to a common scale, that is rescaling the image between 0 and 1. Then comes the shear transformation, which is applying slight distortions to the images. Coming to zooming the image, zooming in on parts of the image to create variations. Coming to horizontal flip, it is flipping the image horizontally to increase the diversity. Then comes reshaping part, it is adjusting image dimensions to match the model's input requirements. Then the next step is a mechanism is set up to stop training when the model performance stops improving, helping to prevent overfitting or underfitting. Then calling the model. So here the architecture of the model is defined, usually involving convolution layers for feature extraction and classification purpose. Then comes building and training the models. So here the model is built using the defined architecture and the training is performed on the process data to adjust the model weights. Then comes the dumping the best model part, where once the model performs optimally, it is safe to be used for the future predictions and further analysis. So this is how we need to do the classification, and then comes the detection part. So here all the libraries which are needed for detection purpose related to YOLO, we will be importing all of them. Then comes exploring the data set. So here the data set is loaded and sample images are explored to ensure they are ready for the pre-processing. So images are loaded into the system. Visualizing, here we have also visualized the images to check for any issues before processing. Then comes image processing part. So here transforming images into a format suitable for YOLO. So here we will be transforming images into a suitable format of YOLO. Then comes defining the class. So here we specify the classes, that is types of disease. Then we declare bounding boxes, that is setting the boundaries around the detected object. Then comes converting image data into a format that model can process. So here we will be converting all the image arrays into NumPy array. After that, we the pre-trained models, that is YOLO models, are loaded and the layers are prepared for fine-tuning. The layers of YOLO network are examined to understand its structure. The final layers are responsible for prediction are extracted. Then comes further image processing. So here we combine images and their labels, that is we append images and the annotations. And then we change the color formats to match YOLO's requirements, that is from BGR to RGB. Then we create a mask for the image to highlight the areas of interest and we resize the image. Then comes data augmentation part. So we increase the data set diversity through random transformations. So we introduce random changes to the images. We apply rotation for variation and other transformations are applied to diversify the images. Then all the images are pre-processed based on YOLO according to the YOLO specifications. And coming to building the model, so here YOLO based model is defined and prepared for training, and the model is trained on the pre-processed data to detect abnormalities in the crop leaves, and the best YOLO model will be saved for the future use in detecting abnormalities. So this is how we need to do the detection. Then moving on to the front end part. So the user interface is built using Flask framework with registration and login functionality supported by SQLite database for user management. And the front end interacts with the model using the data to classify and detect the diseases. And in the front end, users they will be able to upload images of crop leaves for classification and also for detection purpose. The three data sets are used to classify the image and also the detecting the crop disease image. And that given image, it will be pre-processed at the backend. It will be feeded with the trained model for classification as well as detection, and outcome will be displayed through the front end of the application. So this is how we need to implement the project. Now coming to the algorithms used, these are the algorithms trained on all the three data sets for detecting the crop disease, for classifying. So these are the algorithms used for classification purpose. This is the extension model, and this is also the extension model. Coming to the detection part, we have used various versions of the YOLO, which are YOLO V5X6, YOLO V5S6, YOLO V8N, YOLO V9N. So these are the models used for detection of the crop disease. And to implement this project, we need basic requirements, which are hardware and software. Coming to hardware, we need operating system of Windows, processor of 5 and above, RAM of 8 GB and above, and hard disk of 25 GB and above. Coming to software, we need application of Anaconda, primary language of Python, front-end framework of Flask, back-end framework of Jupyter Notebook, database of SQLite 3, and front-end technologies are HTML, CSS, JavaScript, and Bootstrap 4. So this is the overview of the project. Now let us execute. To execute, we are supposed to open the code folder that contains source code file. So let us open the project. So these are the source code folders of the project. Now let us understand each folder. Let's open the application folder. So here in the model folder, we have the algorithm information that is stored in .h5 files, numpy files, pickle files, and that will be loaded into the project code during runtime of the application. And here in the sample folder, we have test cases that are the images that we will be using in the front end for for classifying the crop disease and also for the detection part. And here, this is the static folder. This folder consists of CSS, JavaScript, and Bootstrap files, which are important for the visual appeal. And then comes the templates folder. This folder contains all the HTML pages used in this project. It typically includes files like index.html, about.html, etc., which represent different pages of the website. And this is the app.py file. This .py file contains the information related to front-end logic. It includes code written in Python that handles server-side operations such as processing user requests, interacting with the database, and generating dynamic content to be rendered in HTML templates. This is the signup.db file. This file is a database file which is used to store the user information. So this is all about in the application folder. Now let's get back and open the notebook folder. So these are the files in the notebook folder. So in the cassava folder, we have the data set of cassava. So here it is split into two parts. One is train and the other one is test. So train part, images which are in the train part are used to train the model, and images which are in the test part are used to evaluate the model. Let's open the train folder. So here we have total 5 classes, 5 different types of images where in this folder, we have images related to cassava bacterial blight. So these are the images of cassava where bacterial blight has been affected. And here we have same cassava images where it is affected with the brown streak disease. And here in this folder, we have green mottle of cassava. Similarly, we do have cassava images with their respective diseases. So we will be training this this folder to train the model to classify the crop disease, and images which are in this folder we will be using for evaluating on the unseen data. Let's get back. And here in the plant village dataset folder, so here we have total 40 classes. So total here we have 40 different types of crop diseases, and these images we will be using for training purpose. Let's open images which are here. So here these are the images of apple where it is affected with the scab disease. And here we have images of apple where it is affected with the black rot. Similarly, we have total 38 cases with their respective crop diseases. Let's get back. So here in this folder, we have images split it into train and validation. So images which are in train, we will be using for training and test for evaluating. So here we have total 11 cases. So let's open the bacterial spot. So these are the images of tomato leaves where bacterial spot has been affected. Let's get back. And here are the tomato leaves where early blight has been affected. Similarly, we do have other kinds of diseases along with the healthy tomato leaf. Let's get back. So images which are in these three folders are used for classification purpose. They are used to just classification. And images which are in these three folders, that is YOLO V8, YOLO V5, YOLO V9, they are used to detection purpose. Let us open images which are in YOLO V5. So here also images are split into train and test. So according to classification here, similarly detection also we will be using images which are in train for training part and test images for testing part. Let's open images which are in train. So here we have two more labels, that is two more folders. One is images and the other one is labels. Let us open the images folder. So these are the images of all data sets. So here all the three data set images are combined, that is cassava, plant village, and also the tomato leaf. So all the three data sets are combined and formed into one particular file. They are stored in one particular file. Let's get back. Coming to label folder. In this folder, we have files containing annotations or labels for the objects. That means here we have images, right? So here in the label folder, we have the location of the disease in that image. So let's open. Here, these are the files containing annotations or labels for the objects in corresponding images. So here, each label file corresponds to an image file in the image folder. For example, this first label file, it corresponds to the first image and shows the object locations. That means where the disease is present in that particular leaf. Similarly, for the second image file, we have annotations in this labels. Accordingly, for all the 4,528 images, we have 4,528 label files. How many images we have in the images folder, and that many label files we have in the label folder also. Let's open one file. So let's open this. So this this label files, they follow the YOLO format. So here each line specifies object class index followed by its bounding box coordinates. So this is the object class index and these are the bounding box coordinates. Close this, and similarly we will be having in all these files also. Let's get back. Let's get back. We will be having in the similar way in test folder also where we have images and labels. Let's get back. And here we have YOLO V8, YOLO V9. We do have same kind of images in YOLO V8, YOLO V9 also because each YOLO version requires its own specific type of annotations for training and testing the models. As we are training YOLO V5, YOLO V8, and YOLO V9, we have images in all these three folders. Because having separate folders helps keeping the dataset organized, ensuring that each YOLO version receives the correct annotation it needs to function properly. And here we have 4 notebook files. These are the Jupyter notebook files which contains the combination of code, graphs and outputs all in one place. It allows users to write and execute code in individual cells, which makes it a popular choice for the data science. Here in this notebook file, all these three for the classification and this one is for the, sorry, this last two and the first one are the classification Jupyter notebook files, where in this Jupyter notebook file, we will be loading this cassava dataset and we will be training all the models for detecting the disease of cassava. Similarly, for this Jupyter notebook file, in this Jupyter notebook file, we will be loading this plant village dataset and we will be training all the models for classifying diseases of plant village dataset. Accordingly, tomato also. And here comes the detection part. So here we will be loading images of YOLO V5, YOLO V8 and YOLO V9 and we will be training all the YOLO models individually for detection of the crop diseases. Let's get back. Now let's execute. So open the application folder, copy the path of the code folder from the address bar of the file explorer. I'm copying it. Now open the Anaconda prompt. It is open. Give the command cd, give the space, paste the copied path. Now click on enter. So here if you observe, current directory is changed to the code folder's path. Now compile app.py file. So give the command python space app.py and now click on enter. So this command will execute Python script and perform a runtime check for any syntax errors or logical issues. And after running the app.py file, Flask framework will host the application locally at the default address, which is the local host and the port, unless configured differently. So this is the local host and this is the port. Now copy this local link provided by the Flask framework and paste it into any web browser. I'm copying the link. I prefer Google Chrome, so I will paste it in that. Paste the link here, click on enter. And this is the web page of the project, which is displayed in the browser. This is developed using Flask framework. We need to sign up first. Click on sign up and enter all the registration details if you are registering newly. I have already registered, so I'll directly sign in. Click on sign in and give your username and the password. Click on login. We have logged in successfully and we are redirected to classification page of the project. So here we will be doing classification for all the three individual data sets and we will be doing detection part also. So let's do the classification first for the cassava dataset. Here you can see a choose file button, right? We need to click on that for uploading the images. So here we can see a choose file button, right? Click on that. Let's upload images from the sample folder. So here is a sample folder. As we're doing classification, open classification folder. So we will be classifying diseases of all the three folders. So let's try classifying of cassava. And here are all the different cases. Let's see. First, cassava bacterial blight and whether the application will correctly predict it or not. So let's upload this image. Click on open. It has been loaded. Now click on upload. So here we got the prediction. This is the uploaded image. And here we got the prediction that plant disease type is cassava bacterial blight. So here this cassava plant is affected with the bacterial blight disease. So here we can observe that application is correctly classifying. Now let's upload test case of other folder. We have checked with the bacterial blight. Let's check with the brown streak disease. So let's upload this image. Click on open. Now upload it. So this is the uploaded image. And here we got the prediction that plant disease type is cassava brown streak disease. It is CBSD, that is cassava brown streak disease. So it is correctly predicting this disease also. Now let's try with the other image, that is of green mottle, cassava green mottle it is. So here disease that we will be predicting is green mottle. Now let's upload this image, click on open and now upload it. So here we got the prediction. This is the uploaded image. And here we got the prediction that disease type, that is plant disease type is cassava green mottle, that is CGM. So here cassava green mottle has been detected, that is classified. Now let's try predicting other diseases. Click on choose file. So let's upload images of cassava mosaic virus. So let's upload this one. Click on open and now upload it. So here this is the uploaded image. And based on the uploaded image, here we got the prediction that cassava mosaic disease has been predicted. So here cassava mosaic virus is predicted. Let's upload the last image of this cassava dataset. That is the healthy images. So let's upload this one, click on open and now upload it. So here we got the prediction. This is the image that we have uploaded, and here we got the prediction that plant disease is healthy. So here we got the plant disease is healthy. We're done with cassava. Now let's check with the plant village. Click on plant village. Click on choose file. So get back, open plant village. Here we have total 38 cases. Let's try uploading 5 to 6 and you can check with the remaining one. So let's upload images of corn maize healthy. So let's open it. Let's open this one. Let's upload this one. Click on open and now upload it. So here we got the prediction. This is the uploaded one. And here we got the correct prediction that corn leaf is healthy. Now let's try giving some more images of the plant village data set. Let's get back. Let's give images of this folder where corn northern leaf blight. Let's upload it. Click on open. And here we got the prediction. This is the uploaded one. And here we got the prediction that corn northern leaf blight has been classified. So there is a plant disease. This is the image that we have uploaded. And this leaf is being affected with the corn northern leaf blight. Now let's try uploading some more classes of the same data set. Get back. Let's upload images of this folder. Let's upload this one. Click on open and now upload it. So here we got the prediction. This is the uploaded image, and here we got the prediction that grape ESCA measles has been classified. So this is the grape leaf where it is affected with the grape ESCA black measles. Now let's try uploading some more. Click on choose file. Get back. Now let's upload this image. Click on upload. And here we got the prediction. So this is the uploaded image, and here we got the prediction that plant disease type is orange honglongbing citrus greening. So this is the disease that is affected by the orange leaf. Click on plant village. Now let us upload some more. So get back. Now let's try with this one. Select it, click on upload and click on upload. So this is the uploaded image, and here we got the correct prediction that potato leaf blight has been classified. So this is a potato leaf that has been affected with the late blight. Now let's upload the last image of this folder, I mean data set. So let's upload this image, click on open and now upload it. So this is the uploaded image, and here we got the correct prediction that there is a this is the uploaded leaf image and it is healthy. That is soybean leaf it is, and it is healthy leaf. Now let's do prediction for the tomato. Click on tomato. Click on choose file. So open the tomato folder. So let's upload images which are in this folder, that is tomato bacterial spot. Select it, open it and click on upload. So this image will be pre-processed at the backend. All the images that we upload will be pre-processed at the backend, and then we will be getting prediction by the train model. So train model will be giving us the prediction. So here we got the prediction. This is the uploaded image, and here we got the prediction that plant disease has been classified, and that is tomato bacterial spot. So this is a tomato leaf which has been affected with the bacterial spot. Now let's try with the one more test case of the other class. Get back. Let's try uploading images which are in this early blight folder. Select it, upload it. So here we got the prediction that tomato early blight has been classified. So this is the tomato leaf that we have uploaded, and it is affected with the early blight. Now let's try uploading some more images. Click on choose file. Get back. So let's upload which are in this folder. Upload this. Click on upload. So here we got the prediction. This is the uploaded image, and here we got the prediction that tomato leaf blight has been classified. So this is the tomato leaf that is affected with the late blight. Now let's check with some more classes. Get back. Click on powdery mildew. Select the image. Click on open and now upload it. So here we got the prediction. This is the uploaded image, and here we got the prediction that tomato mildew has been classified. So this is the tomato image which is affected with the powdery mildew. Now let's upload some more test cases. Click on choose file, get back. Let us upload images from this folder. So let's upload this one. So here we got the prediction that plant disease type is tomato spider mites, two spotted spider mite it is. So this is the uploaded tomato leaf and that has been affected with the spider mites, two spotted spider mites it is. Now let's upload some more. Click on choose file, get back. Let's upload images from this folder. Let us upload this one. And now upload it. So here we got the prediction. This is the uploaded one. And here we got the prediction as tomato early blight. So here tomato early blight has been predicted. Now let's upload last image for the classification. Click on choose file, get back, open yellow curl leaf. So select it, click on open and now upload it. So here we got the prediction. This is the uploaded image and here we got the prediction that plant disease type is tomato yellow curl virus. Tomato yellow curl leaf virus has been predicted. So this is how you need to do the classification here. Coming to the classification, classification is just giving the image. The application will just tell what is that disease affected by the plant. It will just tell what is that. It will not spot where the disease is. Now we will be doing detection where that disease will be spotted on the leaf. Click on detection. Click on choose file. So let us open the detection folder. Open the detection folder. So let's upload the image. Let's upload the first image. Click on open and now upload it. So this image will also be pre-processed at the backend and then trained YOLO model will be giving us a prediction. So here this is the uploaded image and here tomato mosaic virus has been detected and a green color bounding box is drawn around the disease of the leaf and it has labeled it as tomato mosaic virus and given the probability score. That is how accurate the detection is. That is how confident the detection is. Let's upload some more. Click on choose file. So now let's upload this image. So here we can observe that gray ESCA black measles of the grape has been detected. So wherever there is a disease on the leaf, it is drawing a bounding box around the disease. You can observe, right? Let's upload some more. Click on choose file. So let's upload this image. Click on open and now upload and detect it. So here if you observe, orange leaf blight has been detected. So it is gray leaf blight. It is gray leaf blight. Wherever there is a disease, a bounding box is drawn around that. Here you can observe, right? So let's upload some more. Click on choose file. Now let us upload this image. So here you can observe that potato early blight has been detected along with its probability score and the class label and bounding box drawn around the disease wherever it is present. Let's get back. Click on choose file. So now let's upload this image. Click on open and now click on upload. So here you can observe that tomato late blight has been detected. So here tomato late blight is detected. Now let's try uploading some more. So now let's upload this image. Click on open and now upload and detect it. So here you can observe that tomato bacterial spot has been detected. You can observe, right? And with that probability scores. So in this way, you can do the detection and also the classification. So classification, it will just tell what is affected disease and the detection, it will detect where that disease is. Now let's check with the graphs. Click on graph. Let's see graphs of cassava. Click on that. So here the first graph we have is the accuracy comparison graph. Here on x-axis we have accuracy score and on y-axis we have all the algorithms that are trained for classifying the cassava disease. Cassava leaves affected with the diseases. So this is the precision score, this is the recall score, this is the FN score. Similarly, let's open the plant village. So these are the models that are trained on plant village dataset for classifying the crop disease. This is the precision score, this is the recall score, and this is FN score. Now let's check with the tomato. So similarly here also we have the scores of all the models trained on tomato dataset. Now let's check with the detection. So click on detection under graph. So this is the precision recall curve graph, and this is the F1 confidence curve graph, and this is the confusion matrix, and this is the precision score graph, this is the recall score, and this is the mean average precision. Now click on sign out. In this way, just by giving images to the application, it will classify if you want classification, and if you want detection, you also have that option too. So this is all about the project.
Thank you for watching video. For more projects, please visit our website www.trueprojects.in. For updates on latest project videos, please visit True Projects YouTube channel and subscribe.